All right, if you're not able to attend lab in person, then one of the things that you miss out on is the pre-lab talk. So this is Dr. L, and I'm gonna give you the pre-lab, very quick talk for the lactase lab. So in this lab, we were studying the activity of the enzyme lactase. Normally, lactase is produced in the small intestine of humans, for example, and it takes the sugar lactose, OSE means sugar, and that's milk sugar, and a lactase enzyme catalyzes the breaking of lactose into glucose plus galactose. Okay, it's a degradation or a decomposition reaction. Now, the enzyme lactase is really great for beginners to work with because it is extremely robust. So it's very tolerant of changes in timing, temperature, and pH and concentration. So it's easy for beginners to work with. But what's tricky about it is that the reactant or substrate lactose plus the products glucose and galactose, they're clear, they're colorless, which makes them very difficult to measure. And if you can't measure things and put numbers to them, then it's hard to study them. So the workaround here that's commonly used is to use a fake sugar called ONPG. And that is so similar to lactose that the enzyme lactase will bind to it at the active site, and it will catalyze the breaking of ONPG into the products ONP, orthonitrophenol, plus galactose. And the great thing about this is that the ONP is yellow. So when you see yellow in the tubes from this experiment, you're looking at a measure of ONP. The more yellow you have, the more ONP you have. The more ONP you have, that's product, and that means you've had more reactions. So the enzyme lactase would have been more active. So the absorbance of yellow color is a direct measure of product formation, which is then telling us how much the enzyme lactase is working. Okay, so that's the basic idea here, is to monitor product formation using spectrophotometry in this particular chemistry. As I showed you in another video, there were 3 kinds of tubes. So there are blank tubes that we set up for this experiment, okay? And my camera does not want to focus on that well, right? There are control tubes, and then there are like test or experimental tubes. And those are the volumes that get parsed into each type of tube, and I showed you that in another video. The total experimental design, right, it's kind of written up here on the board. So you will do 2 experiments, right? An experiment at pH 5 and an experiment at pH 8. And so you'll have your tubes labeled pH 8 blank, then C, 3 C's and 3 T's. The 3 C's are the no enzyme control, and the 3 T's are the tubes where you will have the enzyme in them. So you should see yellow product formation there. The controls are no enzyme controls, so they're just to make sure that we don't see any yellow ONP formed in the absence of lactase. And all of those would be mixed together as I described in another video, and then let to react for 2 minutes, and then the absorbance at 420 nanometers measured on the spectrophotometer. So we'll have the pH 8 experiment, and then we'll also have the pH 5 experiment. So it's 7 tubes each, a blank, 3 no enzyme controls, and 3 with enzyme test vials or cuvettes or tubes, whatever you want to call them. And all of them are measured by spectrophotometry because the concentration of ONP will go up as the enzyme works, and that means the absorbance of light in the spectrophotometer will go up. And that's pretty much it for the pre-lab talk, other than I pointed out the fact that this data obviously sets itself up in pairs nicely, and so therefore we will be analyzing it soon using the same approach as for a previous lab, where we'll put this data into Google Sheets, and we'll do calculate some base statistics and do some t-tests, so we can compare with and without enzyme, and we can compare with enzyme at the 2 different pHs pretty nicely with this experimental design. And that's it.